Hi everyone. In the previous part, we had gone through protein significance and classification. Now, in this part, we will discuss about structure of proteins. Okay? A functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. The sequence of amino acids determines a protein's 3D structure. A protein structure determines its function. So that's why it's very important to learn about the structure of proteins. Why? Because a protein structure determines the function of the protein. Okay, so to understand how a protein gets its final shape or conformation, we need to understand the four levels of protein structure, that is primary structure, secondary structure, tertiary structure, and quaternary structure. So these four types of the structures we are going to discuss in detail. So here, the levels of protein structure, as I told you, primary, secondary, tertiary, and quaternary. So here, primary structure is going to be a linear sequence of amino acid. Secondary is going to have localized regional structures, and tertiary is going to present an overall shape of the proteins, and quaternary protein structure is going to be seen in some interactions between the proteins. So these are all just levels where they are going to occur. I want to mention. So coming to in detail of these structures. So let's see the first one, that is primary structure. The primary structure, the simplest level of protein structure. So primary, obviously we can compare this primary structure as just the alphabets of English language. So where just there are of all individual letters, in the same manner we have this amino acids individually and arranged in the form of a linear structure. So obviously the linear structure of a protein is being considered as primary structure of protein. The hormone insulin has two polypeptide chains, A and B. The sequence of A chain and the sequence of the B chain can be considered as an example of primary structure. So here is a linear structure and here is a linear structure. So these individually being considered as primary structure. The best example is the insulin. And then coming to the secondary structure refers to the locally folded. That means now the alphabets are put together to form the simple words, isn't it? In the same manner, the primary structure of amino acids are put together to form the secondary structure. The most common types of secondary structures are alpha helix and the beta plated sheet. Both structures are held in shape by hydrogen bonds. Remember this one. Here we are going to have the hydrogen bonds. When we go with the tertiary and the quaternary, we may have the increase of these types of bonds. So just remember, okay, which form between the carbonyl O of one amino acid and the amino group of another amino acid. We know that the amino acid structure, one is going to have a carboxylic group and another one is going to have an amino group. So the linkage is between the carboxyl O of one amino acid and amino group H of another amino acid. Let's see the structure in detail. So this is how the secondary structure of amino acid is going to be seen. So if you observe, I told you the alpha helix, so helical structure. So this is going to be the alpha helix structure, and here is, if you observe, this is a beta pleated structure. Pleated is nothing but the sheets, how they are going to be arranged. So beta plated structure, so this is one and this is the two. So this is the alpha helix structure and this is the beta plated structure. Then moving to the next slide, that is tertiary structure. So here the tertiary structure, it obviously goes with the short sentences, or we can uh, I told you that primary structure can be considered as a alphabets, that the single letters, and the secondary structures can be considered as words, and now the tertiary structures can be considered as sentences. So here the tertiary structure is a combination of primary, secondary, and moving to the third one. They are further folded and gives rise to this one. And this is a three-dimensional structure, and majority of the proteins are going to give the three-dimensional structure. The overall three-dimensional structure of a polypeptide is called its tertiary structure. The tertiary structure is primarily due to the interactions between the R groups of the amino acids that make up on the protein. Remember, in the primary structure and secondary structure, I told you that uh, they are going to be held together, which form hydrogen bonds are being formed between the carbonyl group of one amino acid and the amino group containing H of another amino acid. But coming to here, the tertiary structure is going to be formed between the R groups. In the amino acid structure, we are going to have a R, alkyl group or aromatic group, other than the carboxyl and the amino group. So there it is going to have the interactions. And how they are going to interact? They are going to have the several bonds. In the secondary structure, I told you we are going to have majorly by the hydrogen bonds. And now here in the tertiary structure, we are going to have so many types of the bonds are involved in holding the or forming this tertiary structure. One is going to be of your important hydrophobic interactions, then in which non-polar hydrogen R groups are clustered together, okay, and disulfide bonds are there. So here you are able to see the disulfide bonds, and here are the hydrogen bonds, and here are the ionic bonds, okay. So these are all the few different types of bonds which are involved in the formation of tertiary structure of proteins. And then coming to the next structure, that is quaternary structure. When multiple polypeptide chain subunits come together, then the protein attains its quaternary structure. Example is the best example is the hemoglobin structure. Okay, an example of quaternary structure is the hemoglobin. The hemoglobin, we know that it carries oxygen in the blood, and it is made up of four subunits. So two subunits are beta sheeted and two subunits are alpha sheeted. So that's how we are going to have a quaternary structure. Quaternary does not mean of a four. The number of what we call protein subunits are going to be folded together and giving rise to the quaternary structures. So I hope so you understood, but still I want to give you a clear picture or overview of this concept in detail. So let's have a quick review. Look at this. So here is the primary structure that we have discussed, and then secondary structure where we have alpha helix and the beta pleated sheets, and then this is a tertiary structure and quaternary structure. Listen very carefully. This is an important topic, and that's why I'm having a review of this one. So coming to the primary structure, I told you the primary sequence of amino acid in each polypeptide constitute the primary structure. And coming to the secondary, the primary structure, that is this one, is folded, see here, is folded to form secondary structure. So these are of two types as I told you. One is a helical type and the one is a beta pleated structure. The turns of helix and sheets are attached by hydrogen bonds, remember. So here these two are held by this hydrogen bonds. So this is going to be the hydrogen bonds that we have to remember. And then coming to tertiary structure, it gives the three-dimensional structure of two protein, and tertiary structure is the stabilized by several types of bonds. I told you here only hydrogen bonds are involved, but in the tertiary we already discussed a few bonds like disulfide bonds I told you. Then here you can see ionic bonds, hydrogen bonds, disulfide bonds, hydrophobic interactions. So these are all the bonds again I am mentioning here. So what are the bonds that we have discussed in the tertiary structure? One is going to be of, as usual, hydrogen bonds, and then ionic bonds, then disulfide bonds, some covalent bonds, van der Waals interactions, and hydrophobic bonds. So if you see here, these are the linear structure, that is a helical structure, and outer side you're having a beta plated structure. And then coming to the fourth one, quaternary structure, found only in multimeric proteins, where two tertiary structures joins as a subunit. So two tertiary structures are going to be joined and giving rise to the as a subunit, and that's how quaternary structure is going to be. Now just assume that if the primary structure or alphabets, if primary structures are alphabets and the secondary structure are words and the tertiary structures are sentences, so we can consider this quaternary structure as paragraphs or paras. So that's all about the structure of proteins. I hope so you all understood well. Okay, and we will continue the properties of proteins in the next part. Thank you.